Here we are, AP Biology, the chapter 13, Evolution. This is the third and final video in this chapter. We have previously talked about Charles Darwin and his mechanism for the theory of evolution, which is natural selection. We talked about how evolving populations can be studied mathematically using Hardy-Weinberg. And now what we're going to do is talk about some of the additional things that can contribute to evolution. And the first of them is genetic drift. Genetic drift can contribute to evolution because it is a change in the gene pool that happens in a population due to random chance. And so due to probability alone, the frequency of an allele has become altered. And that change in the frequency allele then mathematically is represented in Hardy-Weinberg as being evolution. So one of the examples is in genetic drift happen for, like, example, the founder effect or the bottleneck effect. So here, if we look at our original population, there's green, yellow, and purple, and you can see kind of in what ratio they are. But let's say that we have been on a massive hunting spree and we've killed all of the population except for these three remaining. Well, then we use these three remaining and we say, okay, here's our surviving population. Let's save them and allow them to reproduce. Well, in doing so, we have eliminated and altered the gene frequencies. We have eliminated the yellow. You can tell that we've altered the ratio between purple and green. And now this surviving population represents the founding or the founder effect genetics that then can rebuild a population. But it is forever altered allele frequency-wise from the original population. Now, gene flow is our way of saying that the genes or genetics can change when individuals between a population come in or out. So if migration or emigration happens, if we're bringing genes in or out with us. And that in itself can alter the allele frequencies in a population for Hardy-Weinberg. So what happens is that these individuals that come in or out of a population are the individuals that found a population. The nature that they live in, the environment, is going to kind of give them differential reproductive success. So some of them are going to have traits that allow them to be more reproductively successful, which then turn allows their genes to show up more often generation after generation. One of the problems is that endangered species often have a low genetic variability, and that's because there's so few individuals left in an endangered species that we use them to propagate the population again, but they don't have all the variability that originally happened. One of the major examples is cheetahs, and we have lowered their numbers so low, and unfortunately within the surviving population was a rare genetic disease that shortened their lifespan, but we had to allow them to reproduce to save the population. But now we have allowed a rare allele to become very, very common over time. Now, one of the big ideas is that this is all about reproductive success and how some have traits that are better. When we talk about that, what we're referring to is the variation. Variation is often called, in a fancy way, polymorphism. And that means that we allow for different types of characteristics. So different skin color, eye color, hair color, heights, things like that. Now, populations can also have what's called geographic variation. And that is where whatever inherited characteristics you have kind of happen based on where you are in geography. And so one of the things that can happen is, yes, sometimes we get mutations. So they randomly happen, and let's say you have all these lizards that are black, instantly you get a mutation that allows one to be white. Oh, but what if that white one is reproductively successful? Now the white mutation or the white gene or allele becomes more frequent. Then we're having evolution over time. The other thing that can happen is that sexual recombination. So when we combine sperm and egg, we allow for a greater kind of variation to occur, which allows for more phenotypes or more traits, which then allows for more variation when we're going for and picking traits for evolution. How does this happen? Well, sexual recombination is simply referring to the fact that we have crossing over that occurs during meiosis, which is the process of how we make gametes, our sperm and our eggs. This process we have studied extensively earlier on in the semester. If we need a review, this little diagram does a pretty good job. Otherwise, let's go back and rehash semester 1 material. One of the big things about this is that we can look at evolution And we have some serious health problems that come with this. And the biggest one is antibiotic resistance. Because we live in a society in which people are sick, we give them antibiotics. We're not always able to successfully kill all of the bacteria or kill them target the correct bacteria. And so what's happening is we're focusing a population by giving antibiotics. We're only selecting bacteria that is resistant to the antibiotic we're giving. And then we're allowing that bacteria time to reproduce and multiply, which then causes a problem because when we go to get rid of the bacteria by using antibiotics again, they don't work. Now, one of the big things that happens when we look at variation is we have to go back to Gregor Mendel and his genetics with dominant and recessive. Because we have diploidy, which means we have two versions of every gene, two alleles, that you can have recessive alleles kind of hiding around in a genome even if you don't look like that trait. And sometimes that can result in a heterozygous advantage, which basically says if you carry the recessive trait, you have an advantage over someone who is homozygous dominant, does not have the trait, or someone who is homozygous recessive and exhibits the trait. One of the best examples is sickle cell anemia. Sickle cell anemia, if you carry the trait, you have some sickle blood cells, which is good because if you live in an area with malaria, you're less likely to get malaria. But you don't have sickle cell anemia. You only carry the trait for it. Because if you had sickle cell anemia, you'd have a lot of health problems. Whereas if you are homozygous dominant and have normal blood cells in a malaria area, guess what happens? You're going to get malaria and get sick. So it turns out that you have a heterozygous advantage simply because you are carrying but not exhibiting the recessive allele. Some variations just don't matter. They don't provide any sort of success. One of them is fingerprints. There's no apparent advantage or disadvantage based on what kind of fingerprints you have. It's not used for sexual selection. Like partners don't go up to each other and say, let me see your fingerprint before I make babies with you. That's not going to happen. One of the big things about this is we're talking about passing on your traits. When we talk about that, what we're referring to is your fitness, not your physical health, but what we're talking about is your ability to donate genes to the next generation. So if you reproduce, you pass or donate your genes on to the next generation. That makes you fit or have fitness. Now, one of the things about this natural selection is that it can change the variation that we exhibit three different ways. There's stabilizing, directional, and disruptive. And I think it's easier to understand these three in picture format. So here we have our phenotype, which is fur color of mice. So here's our original population. We have some light, some dark. Most of them fall somewhere in the middle of phenotype. With stabilizing selection, what happens over time is that we favor the phenotype in the middle. So here, notice the population. We've eliminated the light, we've eliminated the dark, and we have made all of the population have the phenotypes somewhere within this middle range. Directional selection says we're going to go in a certain direction with phenotypes. So you can see here's our original population. We've eliminated the lighter colored phenotypes, and now the next generation will just have the darker skin tone. And then third type of natural selection effect that can happen is disruptive. And that's where we're favoring the light and the dark phenotypes. You can see the light and the dark here. And we're actually minimizing the number that falls somewhere in the middle, this kind of general average phenotype. One of the reasons that this can happen is, or one of the reasons disruptive happens, is that sexual selection actually leads to what's called sexual dimorphism, which means you have sexual characteristics that make a boy look like a boy and a girl look like a girl. And that's important because having a boy look like a boy allows for greater sexual selection for him by the females. And so some of the examples are, like in mating, male deer have horns. That is a secondary characteristic. It turns out that having horns or larger horns allows you to fight, allows you to win the fights for the mating rights to females. So they pass on the trait for big horns. Peacocks do their little tail feather display. Again, it's all about having the luck with passing on the traits to the next generation. So the big idea, though, about this whole change thing is it's not perfect, right? We can only work with what genes we've been given. We can only work with what mutations are introduced or what changes happen. And if nature changes, then what traits are selected changes as well. So we have to consider this to be an ongoing process. That's it. Now we're done looking at microevolution. The next chapter is going to look at macroevolution.